Dear student, welcome to EPG Pathshala. Today we will discuss on the module on morphology, life cycle, genetic aspects and diagnosis of Toxoplasma gondii. In this chapter, after the final reading of, we will excrete out some learning points like you can understand and identify the different morphological forms of the Toxoplasma gondii. You can explain the different forms of pathogenicity in different ways, and you can explain the intermediate and final hosts in their life cycle and highlights some symptoms and diagnosis methods of the Toxoplasma gondii. Finally, we will discuss on various drugs and vaccinations for prevention and controlling of the disease. First of all, now we will discuss on taxonomic classification of Toxoplasma gondii. According to NCBI taxonomic database, Toxoplasma gondii come under the kingdom Alveolata and phylum Apicomplexa, class Coccidia, and genus Toxoplasma, species gondii. The word Toxoplasma is comprised of two words like toxon and plasmid. The both words are originated from Greek word. The former word toxon means wow and later plasmid means form. Therefore, the original Greek meaning of the word Toxoplasma is wow-shaped organism. Although Toxoplasma gondii is an obligate intracellular protozoan parasite, its distribution is throughout the world. The disease Toxoplasmosis is caused by this parasite in all the warm-blooded animals. Toxoplasmosis also leading the cause of abortion in sheep and goats. Toxoplasma gondii exists in three forms, and all forms are infectious. The form Trophozoite or Tachyzoite found in groups or clones, and the Bradyzoite found in tissue cysts, and the Sporozoite found in oocysts. All the three stages are linked in a complex life cycle and necessary to maintain the parasite. They can undergo sexual or asexual reproduction depending on the host. Both the Trophozoite and Bradyzoite stages are represented by the schizogony, and the Sporozoite stage is formed by either gametogony or sporogony. All of these three forms can occur in the domestic cats as well as in other felines, which are the definitive hosts of these sustains to both the schizogony and gametogony. Although the two forms like Trophozoites and Bradyzoites also exist in other warm-blooded animals, including birds and humans, which are the intermediate host for them. In the first Trophozoite stage, the Trophozoite is a oval-shaped parasite form of Toxoplasma gondii, which an anteriorly conoidal and posteriorly rounded end. Its dimension is 2 microM to 4 micrometer wide and 5 micrometer to 8 micrometer long, and is stained well with both giemsa and white. It is a rapid growing stage observed in the early stage of infection. Frankel in 1973 replaced the term Trophozoite by the term Tachyzoite. Trophozoites have not to show any sign of motility, means like flagella, cilia, pseudopodia, but they show movement by flexing, gliding, rotating and undulating. Trophozoites also enter into the host cells by the phagocytosis or through the active penetration into the plasma lemma of host cell. In Trophozoites, the nucleus is placed towards the central part of the cell, and having a centrally placed nucleolus and chromatin clumps. The anterior typical epicomplexan organelle present in the trophozoite is the apicoplast, which is a non-photosynthetic and homologous to chloroplast of plants. It has its peculiar circular genome and synthesizes its own specific proteins. At the anterior end of trophozoite, the polar ring 1 is present, which is an electron dense structure formed by the thickening of inner membrane complex. It is an essential component of the parasite for its mobility and penetration into a host cell. In addition, from polar ring 2, approximately 22 members of subpellicular microtubules originate and run longitudinally to an almost entire length of the cell, just beneath the inner membrane complex. These microtubules are organized in a mild spiral and similar to a rib cage. The 8 to 10 club-shaped organelles present between the nucleus and anterior tip are called as rhaptres. The rod-shaped structure mostly occur at the anterior end of trophozoite is called as micronemes. The exact function of rhaptres, conoid, micronemes and micropores are not completely understood, but perhaps they are linked with penetration into the host cell as well as for making an intracellular environment which would be appropriate for the growth and development of the parasite. After entering into the host cell, motile and quickly multiplying trophozoite is responsible for increasing its population in the host and gets converted into an ovoid shape, which is surrounded by a parasitophorous vacuole. This vacuole seems to be derived from host as well as parasite cells. After penetration, the tubulovascular membranous network develops inside these parasitophorous vacuoles, and then initially trophozoites spread into whole body by a blood stream. But further invasion and growth rate depends upon the Toxoplasma gondii strain, host cell type, and or the development of humoral antibodies. Due to those reasons, if the host cell does not extend to support for the growth of trophozoite, it will be rupture. The second phase of Toxoplasma gondii is Bradyzoite. The Bradyzoite, a crescent shaped phase of the parasite Toxoplasma gondii, was also coined by Frankel in 1973. It is a slow replicating stage developed within the cytoplasm of the host cell and make tissue cyst wall. Hence, Bradyzoites are also called as cystozoites. If the tissue cyst contains only a few bradyzoites, it may be smaller in diameter, like 5 micron, while the older ones may contain from hundreds to several thousands of bradyzoites divided by endodiazony, which may be larger in diameter, like 10 to 200 micrometer. The tissue sheets of bradyzoites are more common in muscular and neural tissues, including the eyes, brain, cardiac and skeletal muscles. They are less prevalent in visceral organs such as lungs, kidney and liver. The intact tissue cysts of Bradyzoites perhaps do not cause any injury in the host. It can persist throughout the life without causing to any inflammatory response in the host cells or tissues. The light microscopy study revealed that the Bradyzoite form a parasite seems to be structurally similar to the Trophozoite form. But ultrastructural studies revealed that the Bradyzoite has a nucleus at posterior end and contains numerous amylopectin granules. Mostly in aged Bradyzoites, the contents of rhabdries are generally electron dense, but in younger tissue sets may have labyrinthines. Most of the Bradyzoites possess 1 to 3 rhabdries which are looped back on them. Again, Bradyzoites mark the chronic phase of infection and resistant to low pH and digestive enzymes during stomach passage. The Bradyzoites within the cyst stains strongly positive with periodic acid shifts or pass stain. The third forms of Trophozoite are Sporozoites. The Sporozoites is a parasitic stage of the parasite Toxoplasma gondii, which is surrounded by an extremely resistant thin wall known as an oocyst. The oocyst is an ovoid or spherical in shape, measuring approximately 10 to 12 micrometer in diameter and possesses the sporoblast. The oocyst is non-infectious before sporulation. The unsporulated oocysts are subspherical to spherical and sporulated oocysts are subspherical to ellipsoidal. But when the definitive hosts like cats infected by the ingestion of either oocysts or tissue cysts, it can shed millions of oocysts per day in feces, but the freshly passed oocysts are not infectious. The oocysts become infectious only after the development in water or in soil for few days according to the availability of temperature and aeration. During this sporulation state, the sporoblast divides into two ellipsoidal sporocysts without stida bodies, measuring 6 to 8 micrometer in diameter. Each oocyst has two ellipsoidal sporocysts and each sporocyst contains 4 sporozoites. Now, sporozoites are the infective stage of the parasite and ultrastructurally 2 by 6 to 8 micrometer in size, with a subterminal nucleus along with plenty of micronemes, rhabdries, amylopectin granules and without any crystalloid bodies. The sporoblasts possess a strong resistance against the adverse environmental conditions and they can remain infective in water or soil for a year. When the infective oocyst is ingested by another definitive host, it releases sporozoites in the intestine of the host, which initiate infection. Now we can discuss on some hosts. The definitive host is a cat or domestic cats who pick up the organism from eating infected rodents, and oocysts producing sexually developed or development during the life cycle of Toxoplasma gondii. Humans are also an intermediate host, and the parasite complete his asexual life cycle in the humans and produce motile phase like Taxozoites and non-motile phase like Bradyzoites. Now we will discuss on life cycle of Toxoplasma gondii. The life cycle of Toxoplasma gondii has completed within a wide variety of hosts, especially in all warm-blooded animals. The life cycle contains of two reproductive phases like asexual and sexual. The sexual reproductive phase occurs only in the cat or feline families members, and the asexual reproductive phase of the parasite occurs in both intermediate like birds or mammals as well as final or definitive hosts like cats. The sexual phase are also called as intestinal phase and occurs only in definitive hosts like cats and produce only oocysts. An asexual phase also called as extraintestinal phase and occurs in all animals that in fact definite and intermediate hosts. And Taxozoites are produced and eventually Bradyzoites are produced during the asexual phase. In the sexual reproductive phase, the sexual reproductive phase of Toxoplasma gondii takes place in the definitive hosts intestine like in domestic cat. The domestic cat acquire the infection by ingesting any of the infectious stages like Trophozoite, Bradyzoite or Sporozoite of Toxoplasma gondii due to feed of infected prey. First of all, the parasite infects the epithelial cell of the ileum and then starts the asexual development in a series of distinct morphological schizont stages that exhibit particular division of characteristics. Then the gamete formation is likely initiated by merozoites released from schizont stage approximately 3 days of the post-infection of definitive host. The female macrogamete contains numerous organelles, while the male microgametes harbors up to 22 microgametes. The male microgametes possess a top and perforatorium organelle and flagella which they use to swim, penetrate and fertilize mature female macrogametes to form zygotes. Now several layers of cyst wall are formed around the parasite. Infected epithelial cell rupture and oocysts are released into the intestinal lumen and then excreted following defecation, and sporulation subsequently takes place in nature. Now, within 1 to 5 days of post excretion of sporulated oocysts containing 2 sporocysts harboring 4 sporozoites each, and finally this stage of parasite will be ready to start a new cycle. In the asexual reproductive period of Toxoplasma gondii takes place in intermediate hosts like birds or mammals, as well as in the definitive host also like cat. During this phase, oocysts or tissue cysts ingestion with edible food or prey is main path of infection in the targeted hosts. Cats are less sensitive to tissue cyst or oocysts infection than human or birds. The pre-patent period depends on the only ingested form but not on the dose level. The resilient cyst wall is subsequently degraded by the gastric juice or proteolytic enzymes present in the small intestine and stomach. The sporozoites and or bradyzoites are released in the small intestine, which thereafter penetrates into the goblet cells and enterocysts of the intestinal epithelium. Then these sporozoites and or bradyzoites carry to lamina propria, where they proliferate, producing the fast replicating trophozoites. The trophozoites are potent to infect and multiply in almost all nucleated cells of mammals or birds through its rapidly intracellular growth and spread in all over the body of the host through circulation within 12 to 16 hours, leading to cause parasitemia, which is an acute phase of toxoplasmosis. After 5 to 10 days of ingestion, the trophozoites disseminate to peripheral organ and muscles. Once the host is able to produce a specific immune response, the parasites transform from trophozoite to bradyzoite form and insist themselves in various tissues. Although the oocyst induced cycle is similar to the bradyzoite induced cycle, following the ingestion of infected meat or prey, but the sporozoites are more infectious to intermediate hosts compared to the bradyzoites. Due to normal immune response and transformation of trophozoites into bradyzoite cysts, limit the acute stage of disease and establish a chronic infection. These cysts can persist submassively in the body of host for many years until the host is eaten by a predator or a new cycle starts. Now we will discuss on genetic aspects. All the stages of Toxoplasma gondii except unsporulated oocysts have a haploid nucleus along with the whole genome of 14 chromosomes. The genotypic classification of Toxoplasma gondii is performed by analyzing several polymorphous loci. More than 50 markers have been described, and most of the used ones in genes coding for major antigens and microsatellites. Based on multiple allele analysis, most of the isolates can be grouped into three major genotypes like type 1, type 2 and type 3. These three major lineages differ only in 1 to 2% at the DNA sequence level, though the genetic transfer occurs between those major genotypes, resulting different combinations of alleles. Till today, a number of methods have been established for the classification of Toxoplasma gondii isolates. One of the first methods to be used for strain typing was multi-locus enzyme electrophoresis or MLE. This method used numerous polymorphic enzymes and permitted to the group of a majority of isolates into three major zymodems like Z1, Z2 and Z3. Multi-locus enzyme electrophoresis was a highly precise method but required a large number of parasites. Therefore, new methods based on the PCR techniques and parasites genomic DNA were developed, such as polymerase chain reaction, restriction fragment length polymorphism, or PCR RFLP, sequencing and length polymorphism of microsatellites. PCR RFLP analysis of definite gene loci is widely used for Toxoplasma gondii genotyping. This PCR RFLP analysis of multiple loci also permits to classify the isolates into three major groups like type 1, 2 and 3. PCR RFLP has its limitations due to variation in the restriction enzyme sites and may be the lack of many polymorphisms. Then direct sequencing has an advantage of revealing the complete genetic variations and also detects the more complex and huge genetic diversity, which is more important for the discovery of new exotic strains, but its high cost is the main drawback. So nowadays the microsatellite multiplex assay has been established that permits to compare the both isolates and also determine if they are genetically identical or different, which can be useful for identifying the common source of infection. The disease Toxoplasmosis is spread through blood and lymph system. This disease is of two types on basis of the mode of transmission. One, acquired toxoplasmosis, and second, congenital toxoplasmosis. Transmission may be required the following ways, like transplacental, which may cause severe as well as lifelong disabilities in the sick newborns, ingested oocysts defecated by the infected cat through its feces, tissue cyst consumption by undercooked or uncooked meat. Transmission also occurs through transfusion of blood or blood products, organ transplantation like kidney and liver, intake of unpasteurized goat milk, and eating to unwashed raw vegetable and fruits. Symptoms of the disease have no any sign or symptoms in early stage, but some pupils develop some sign and symptoms similar to those of flu, like body aches, swollen lymph nodes, headache, fever, fatigue, etc. If people having weakened immune system develop more severe signs and symptoms like headache, confusion, poor coordination, sneezing, lung problems that may resemble like tuberculosis, and other like ocular toxoplasmosis. If we see in a newborn baby which are the high risk of toxoplasmosis early stage results in stillbirth or miscarriage. Therefore, infants who survive may have problems such as intracerebral calcification, chorioretinosis, hydrocephaly, microcephaly, convulsions, mental retardation, cardiomegaly, etc. This disease will exactly diagnosed by different diagnostic methods, because a number of direct and indirect techniques have been developed for the diagnosis of toxoplasmosis along with on a specific sensitivity. According to history of the infections, a different set of methods also used to establish the final diagnosis. Therefore, we will discuss some diagnostic methods like one, histopathological analysis, Sabin-Feldman dye test, detection of antibodies, direct agglutination test, and detection of DNA by PCR or polymerase chain reactions. In histopathological analysis, the demonstration of tachizoids in tissue sections or smears of the body fluid established the diagnosis of the acute infection, but it is difficult to determine the tachizoids in conventionally stained tissue sections. The immune peroxidase technique or immune peroxide techniques, which is based on the use of anti-sera to Toxoplasma gondii, has been probed sensitive as well as specific demonstration, and it is also applicable to the unfixed or formalin fixed paraffin embedded tissue sections. In the second test, Sabin-Feldman dye test SFDT was also developed by Albert Sabin and Harry Feldman in 1948. The first novel serological basic methylene blue dye based test for the laboratory diagnosis of Toxoplasma gondii infection. This is a highly sensitive and specific test, but no evidence found for false result in humans true till date. Therefore, it has been still considered as gold standard test. SFDT detects the presence of anti-Toxoplasma gondii specific antibodies, total immunoglobulin as dead and live. In the third test, like detection of antibodies, the Remington, Miller and Brownlee in 1968 proposed this test for the first time in order to detect antibodies in infant serum or in cord blood for diagnosis of the congenital toxoplasmosis, as this IgM antibodies unable to cross the placenta, whereas IgG antibodies can. Detection of immunoglobulin IgM is an auxiliary test to decide whether the infection is acute or it has been acquired earlier. When the IgM serological reaction is positive in an asymptomatic test of host, the test is basically based on the strength of an ionic binding between antibody and antigen. There will be a greater strength of ionic binding when an antibody is produced from an older infection as compared to the recent one. So, this IgM antibodies detection test is not flawless for the critical interpretation. Now, we can go the direct agglutination test. This test was developed initially by Fulton in the year 1965. Later on, it was improved by Desmonds and Remington in 1980 and Dewey and Desmonds in 1987, who called it the modified agglutination test or MAT. Serologically diagnosis of toxoplasmosis in any host is possible due to the development of an uncomplicated and simple direct agglutination. This that is very simple and easy, and it requires no special apparatus or conjugates. This mat is widely used for the diagnosis of toxoplasmosis in animals also. Now, detection through polymerase chain reaction of DNA, we will discuss the first report of work in 1989 for the detection of Toxoplasma gondii DNA from a single Taxozoite using the V1 gene in a polymerase chain reaction or PCR. Nowadays, various PCR tests have been developed using different gene targets. Overall, this technique has established as a precisely valuable for the diagnosis of clinical toxoplasmosis. After the diagnosis, we will go for treatment. The toxoplasmosis treatment has not yet been a victory of modern medicine. The combination of anti-malarial drugs like pyrimethamine and sulfonamide provides a synergistic effect. The spiramycin is the preferred drug at many places for the treatment of acute toxoplasmosis during pregnancy. It dismisses the risk of mental fetal transmission by about 60%. If the fetus gets infected, the combination of pyrimethamine, sulfonamides and folinic acid is given during the whole period of pregnancy. Clindamycin, which also treats the toxoplasmosis, but this is given to those patients especially who showed an allergy to sulfonamides drug. And vaccination of sheep with the live cyst-less strain of Toxoplasma gondii is commercially available and decrease the neonatal mortality in the lambs. But there is no suitable vaccine for human use till the date. Now we will summarize the module in the following specific points. First, Toxoplasma gondii is a obligate intracellular protozoan parasite cause toxoplasmosis in all warm-blooded animals. It has three major forms: trophozoite, bradyzoites and sporozoites. And these forms create pathogenicity in different ways and has its specific genotyping like type 1, type 2 and type 3. Because all genotypes have specific pathogenicity and prevalence and life-threatening diseases in different ways, especially in immune compromised patients, newborns and fetuses, etc. The toxoplasmosis we can diagnose through the different methods, and finally the most precious method is RT-PCR and PCR, and the vaccination only developed for sheep and goats but not for men till today. Thank you.